people, welcome to Molecular Biology and Biotechnology with Lucy. Thank you so much guys for watching my videos, and I want to remind you, if you have not subscribed, please do subscribe and then share and comment with your friends. And today we are talking about DNA, the blueprint of life, and walk with me together in this exciting journey of discovering what is this DNA that is talked about all over across even non-scientific fields, because without it actually there is no life. So welcome so much to this lesson. DNA stands for deoxyribonucleic acid. It's a polymer of repeating subunits and monomers called nucleotides. The nucleotide has three parts. Phosphate has a 5-carbon sugar, deoxyribose, and a nitrogenous base can be A, T, C, or G. And these are bonded together by covalent bonds there and a covalent bond there. Nitrogenous bases in DNA are 4: adenine, guanine, cytosine, thymine. And they are in two categories: purines and pyrimidines. Purines because they have a 6-membered ring fused together with a 5-membered ring. Pyrimidines because they only have a 6-membered ring. The DNA structure is a double helix that resembles a ladder, which has two sides, which we call the backbone, made up of sugar, phosphate on both sides, sugar, phosphate, and the middle part, which we call the rung of the ladder, comprising of base pairs. DNA has direction. You can see here 5' to 3'. That's a direction. And the direction comes from the position of the carbons. The 5th carbon here, we usually use the sugar starting from oxygen. This is carbon number 1, carbon number 2, carbon number 3, carbon number 4, and finally carbon number 5. That gives it direction of 5'. And on the other side, carbon number 1, carbon number 2, carbon number 3, where we have a hydroxyl group, and that gives it 3. And on the other side, we can see that it is running the opposite side, so they are antiparallel, 5' to 3'. The base pairs have a complementary rule. That is, we have a purine binding to a pyrimidine. So adenine and guanine are purines, thymine and cytosine are pyrimidines. Now, adenine binds to thymine using a 2-hydrogen bond, and guanine to cytosine using a 3-hydrogen bond. So we can be able to fill up our diagram. This has a 6-membered ring only, meaning it must be a pyrimidine. And because there are 2 bonds, this is thymine, and it binds to adenine. And here we have 3 bonds, and this is having 2 rings. This is a purine. That is guanine, and it binds to cytosine. And this is cytosine, this is guanine. And this is a 2-membered ring, 2 bonds. And you see this is a purine, so this is adenine, and it binds to thymine. For you to remember the complementary base pair rule, we have a mnemonic. We can say at guidance center, A binds to T, and G binds to C. So DNA, we've said, is a double helix, as seen in this diagram. It was proposed by James Watson and Francis Crick in the 1953s. So let's have a refresher. In the diagram, what substance is represented by letter X? Here is our X. Remember, we said that this is the backbone and this is the backbone, and the backbone is made up of a sugar molecule and a phosphate group. So X cannot be ribose, neither can it be deoxyribose. It is a phosphate and it's not adenine. So it is a phosphate. Now write the sequences in the bases example below. This one is reminding us about the complementary base pairing. So A, we said binds to a T, T binds to an A, T, TA, CG, AT, TA, GC, CG, C, C, and C. So DNA has different forms because the helix conforms to different geometries, and the main one is called balanced DNA. It's the most frequent. It's a right-handed helix, and a turn has 10 base pairs. It can also change to other forms in different conditions, like to right-handed form in a short distance. We'll be looking at this when we talk about gene expression. The other form is called A or alternate DNA. It's also right-handed. A single turn has 11 base pairs. It is the most common when DNA is dehydrated and also when there is protein binding. The other form is zigzag DNA. This one is left-handed, and a turn has about 12 base pairs, and it forms a zigzag pattern because of alternating purine and pyrimidine. Let's say a purine A and pyrimidine C. So AC, AC. If you have such a sequence, then it is likely to form a zigzag pattern, and it can be found at the start site of a gene. And again, it is involved in gene regulation, which we'll be coming across. We have other forms of DNA, cDNA, ddNA, eDNA that are a bit rare. And here we have a diagram representation of the different forms, both in 2D and 3D format. So some DNA facts that we have actually come across in our previous lesson. In eukaryotes, the DNA is linear, and the majority is found in the nucleus. That's the nuclear DNA. But we have extrachromosomal, which is circular, and this is found in the mitochondrial organelle called mitochondrial DNA, or chloroplast DNA in the chloroplast organelle in plants. In prokaryotes, we have DNA in circular form, and it's not enclosed in a nucleus. It's called a nucleoid. Here it is. It's a bit folded, and it also has extrachromosomal, which is called a plasmid, and it's also circular. Now, when we talk about all the DNA, for example, in a prokaryote, we have the nucleoid and the plasmid. That totals up DNA in the organism, and we call it the organism's genome. And the same in eukaryotes. So let's review what we now know about DNA. So DNA stands for deoxyribonucleic acid. What's the shape of DNA? Double helix. Who established it? James and Francis. Adenine pairs with thymine. The sides of the ladder are deoxyribose and phosphate. Guanine pairs with cytosine. Two sides are held together by hydrogen bonds. DNA is composed of repeating units called nucleotides. And why do we have different types of DNA? The sequence of bases, like in the zigzag format, the environment, like the A form, and as they all try to conform into limited space, they take different forms. We have a take-home exercise. Using a table, differentiate between the major forms of DNA. Around 20 marks, so get at least 8 differences between them. So coming up in our next lesson, we'll be talking about chromatin, chromosomes, and genes. Thank you very much.